Students, we know that based on monohybrid cross observation, he proposed two laws. One is law of dominance, another one is law of segregation, right? Which are collectively called the laws or principles of inheritance. Now the question is whether these two laws are universally accepted, whether every organism is following this one. It is common to all the organism. Answer is no. The second one, that is law of segregation, is universally accepted. Okay, it is seen in almost all the living organisms, but not the first one. Which one? Law of dominance. Remember that the law of dominance is not acceptable all the time. There are some exceptions. There are organisms which do not obey, they do not show the law of dominance. Clear? So it is not accepted by all, but which one is accepted? Law of segregation ನಾವು ಎಲ್ಲ ಆರ್ಗ್ಯಾನಿಸಮ್ ಕಾಮನ್ ಆಗಿ ಫಾಲೋ ಮಾಡ್ತಾ ಇದೆ, ಆದರೆ law of dominance is not accepted. The phenomena is not common in all organisms. So write down the next heading: deviations to Mendel law or exceptions to Mendel law. You can write down there are few organisms which do not show the Mendel's laws all the time. Certain exceptions for it include number one, write down, there are many. Number one, incomplete dominance. Number two, co-dominance and multiple alleles. Co-dominance and multiple alleles. Number three, pleiotrophy. And number four, polygenic inheritance. So in our next 2 to 3 sessions, including this one, in the next 2 also, I am going to stick on to which one? The deviations or exceptions to Mendel's law. Throughout, totally including this one in the 3 sessions. Remember that what we are discussing, these 4 concepts, especially out of the 4, the first 3 are very important, which do not show or do not obey the law of which one? Dominance. I am still in the monohybrid cross, okay, where I am discussing exceptions to Mendel's law of dominance. In that, the first one is incomplete dominance. Let me first begin with the incomplete dominance. Write down the heading: incomplete dominance, or it is also called partial or blended partial dominance or blended inheritance. Mendel said there is no blending in the law of segregation, right? But in general he speaks about that, but here there is blending. We will see what exactly that we will see. So I am discussing about incomplete dominance phenomena. I will dictate the notes. Listen to me carefully. This concept was given by one of the rediscoverer of Mendel law. There are 3 personalities. Can you recall them? They include Hugo de Vries, Karl Correns and Erich von Schemack, right? Among them, Karl Correns. Karl Correns thought of repeating Mendel's monohybrid cross. What you studied in my last session, he also thought of doing it. But what he did is, he selected, instead of Pisum sativum, pea plant badalu, he thought of doing the same monohybrid cross experiment what we have done in the last class, right? In a different or another plant. Which is that? Which plant he selected? The name of the plant is dog flower plant. Okay, why it is called dog flower, whether it is related to dog, because when you press that flower from its lateral surface, okay, it will open up which look like a mouth of the dog. That's why it is called dog flower plant. No other relationship with the dog. Okay, or it is also called snapdragon, or it is scientific name is Antirrhinum species. So the dog flower or the snapdragon, also called as scientific name Antirrhinum majus, or simply I call Antirrhinum species. Instead of pisum, he has done it in the dog flower plant. So he is doing which one only? Monohybrid cross only. So how many characters he has to select? One character. He selected flower color as a character. Okay, and flower color in that plant, in the dog flower plant, was available in two traits. They are, one is the red flower, another one is the white flower. Red flower and white flower. He started his simple experiment, the same thing that you have already written in the monohybrid cross. You can practice simultaneously. Write down parents. Which one? True breeding. Red flower plant, he crossed with which one? True breeding white flower plant, the same monohybrid cross. Okay, now these two plants are true breeding. That means they are genetically pure, right? So what is their genotype? Capital R, here small r. What is the first step in reproduction? Monohybrid cross ಏನು ಬರ್ದಿದ್ದೀರಿ, ಸೇಮ್ ಥಿಂಗ್ ಅನ್ನ ಇನ್ನೊಮ್ಮೆ ಮಾಡ್ತಾ ಇದ್ದಾನೆ. Formation of gametes. The gametes formed by this plant all contain capital R. The gamete formed by the white flower plant all had what? Small r. Clear, no? Now gametes are ready. It is self-pollination or cross-pollination? Cross-pollination and obtaining which generation? F1 generation. The F1 plant, what he got, it had the genotype capital R, small r, and one thing is clear, it is a what? It is a hybrid plant. Leave that. The genotype is this. It is a monohybrid cross, alwa? So what is the phenotype? Idara character enu? Its a character should be what? Red color, right? No, but Correns was surprised because he was following Mendel. Mendel ಹೇಳಿದ್ದಾನೆ ಮಾಡ್ತಾ ಇದ್ದಾನೆ. Only ಒಂದು ವ್ಯತ್ಯಾಸ ಏನು ಕೇಳಿದ್ರೆ he selected a different plant, right? That is the dog flower. But ಇಲ್ಲಿ ಯಾವ character ಬರಬೇಕಿತ್ತು? Red ಬರಬೇಕಿತ್ತು, because red was the dominant one. We know that. Surprisingly, he didn't got red. At least white ಆದ್ರೂ ಬರ್ಬೋದಿತ್ತಲ್ವಾ? Not even the white. He got which one? Pink. This pink was, right, neither resembling red nor resembling white. It was in between the two. ನೀವು ರೆಡ್ ಮತ್ತು ವೈಟ್ ಎರಡು ಪೇಂಟ್ ಅನ್ನ ಮಿಕ್ಸ್ ಮಾಡಿದ್ರೆ ಅಲ್ವಾ, you will get which one? Pink. Shall I repeat? The progeny what he got in the F1 generation was not resembling any of the parent, neither red nor white, but it was in between the two. ಅಲ್ವಾ, ಇಟ್ ವಾಸ್ ಇನ್ ಬಿಟ್ವೀನ್ ದ ಟು. ಎರಡು ಕಲರ್ ಅನ್ನ ಪೇಂಟ್ಸ್ ಅನ್ನ ಮಿಕ್ಸ್ ಮಾಡಿದ್ರೆ ಹೇಗಿರುತ್ತೋ ಆ ರೀತಿ ಕಲರ್ ಇತ್ತು. ಹಿ ವಾಸ್ ಸರ್ಪ್ರೈಸ್. ಸೊ ಆಸ್ ಐ ಆಮ್ ಗೆಟಿಂಗ್ ಹಿಯರ್ ಪಿಂಕ್ ಕಲರ್, Correns ಥಾಟ್, ವಾಟ್ ಹ್ಯಾಪನ್ ಟು ರೆಡ್ ಅಂಡ್ ವೈಟ್? ಲೆಟ್ ಮಿ see it. Red and white, what happened? To check that, he thought of obtaining which generation? He thought of obtaining F2 generation. How he performed F2? By self-pollination of the F1 plants. Okay, already you know the genotype. What is that? Capital R, small r, capital R, small r. The gamete produced by these plants are same type or different type? Different type. 50% gametes carry capital R, remaining 50% gametes carry small r. Gametes are ready. What is the next step? Next step is checking the possible combination, that is with generation F2. How we will check F2? F2 can be checked with the help of plotting a graphical representation called checkerboard. Define: it is a graphical representation used to check all possible genotypes and phenotypes in a genetic cross, right? Who proposed this? Reginald C. Punnett. I am repeating this definition for the fourth time. If you would have practiced along with me, it would have been perfect to you. Anyhow, I hope you have done it. Okay, gametes are ready. Let me see their possible combination. You know, again, need not any explanation. If these two get fertilized, genotype is capital R, phenotype is what? Red. What about here? Capital R, small r, it is what? Pink. Capital R, small r, pink. Small r, white. In the F2 generation, along with the pink flower, he got which one also? Red as well as white, and he concluded that it is because it is not obeying Mendel's law of dominance. Here something different had operated, and he called that something different phenomena, which was neither resembling the character either of the parents, but it was in between the two, ibbara middle ittu. He called it ಪಾರ್ಷಿಯಲ್ ಡಾಮಿನೆನ್ಸ್ ಅಂತ ಕರೀತಾನೆ. ಅಥವಾ ಎರಡೂ ಕಲರ್ ಮಿಕ್ಸ್ ಆದರೆ ಹೇಗಿರುತ್ತೋ ಹಾಗೆ ಇರೋದ್ರಿಂದ ಬ್ಲೆಂಡೆಡ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಂತ ಕರೀತಾನೆ. ಆರ್ ಹಿ ಸೆಡ್ ಇನ್ಕಂಪ್ಲೀಟ್ ಡಾಮಿನೆನ್ಸ್. The red color is not completely dominant over what? White color. It is incomplete dominance. Is that clear? Now let us see. In this case, I will write here, what is the phenotypic ratio? As it is a monohybrid cross, right? The Mendel's monohybrid cross phenotypic ratio is 3:1. What about here? Here we are getting red flower, pink flower as well as white flower in the ratio out of the 4, right? 1 is red, 2 are pink, 1 is white. Mendel's monohybrid cross phenotypic ratio was 3:1. You are getting the same here? No, it is 1:2:1. Let us see the genotypic ratio. So what is the genotypic ratio? I hope you have calculated. The 3 different genotypes are capital R, capital R, small r, small r, 1 capital R, 2 capital R, small r, 1 small r. In terms of phenotypic ratio as well as the phenotype that you are getting in the F1 generation, right? It is not obeying law of dominance. It is an exception or a deviation that Correns called it as what? Incomplete dominance. Then one more question, how you will define incomplete dominance? It is a phenomena in which, listen to me carefully, okay, otherwise you can write down also. Once you complete up to genotypic ratio, next line write down, it is the phenomena in which the progeny in bracket F1, the progeny does not resemble either of the parents. EITHER does not resemble either of the parents and was in between the two. This is the standard definition which was even asked in the year 2018 board's exam. 1 mark question. Don't simply write one is right not completely dominant over the another is called incomplete dominance. No, you will not get marks. Focus on the definition of incomplete dominance. It is the phenomena in which the progeny plant or the F1 plant does not resemble either of the parent. The pink color is not resembling either red right? Nor the white. Does not resemble either of the parent and was in between the two. ಇಬ್ಬರ ಮಿಡ್ಲ್ ಅಲ್ಲಿ ಇದೆ ಅಲ್ವಾ, ಎರಡು ಕಲರ್ ಮಿಕ್ಸ್ ಮಾಡಿದವ ಹೇಗಿದೆಯೋ ಆ ರೀತಿ ಇದೆ. was in between the two. Sometime what you will do, does not resemble either of the parents, full stop. That is also wrong. You have to complete and was in between the two. Next line, write down. Next line. Next line. Example. Which is the example? Dog flower plant or snapdragon or antirrhinum. At least you have to write the dog flower or the snapdragon. No need to write antirrhinum. That is not given in the NCERT. Any of the one name that you have to write in the exam. Okay. Next line, write down. When a true breeding red flower plant with genotype capital R was crossed with true breeding white flower plant having genotype small r, full stop. Continue the same line. He obtained pink flowered plant with genotype capital R small r in F1 generation. Next line. It was not resembling either of the parents. It was not resembling either of the parents because allele capital R was partially dominant over, not completely, capital R was partially dominant over allele small r. Partially dominant over allele small r. Next line. When he self-pollinated F1 plants, in the F2 generation, along with the pink flower, he obtained red and white flower plants also. Along with pink flower, he obtained red and white flowered plants also. Okay, that also he obtained. Next line. Red, pink and white flower plants were obtained in 1:2:1 ratio with the genotype capital R, capital R small r and small r with 1:2:1 ratio. Okay, so this is the flow chart. Up to you have to write here and then you have to write what? Its explanation. Take a note separately. Note. The concept of incomplete dominance was given by, in the note separately write down, the concept of incomplete dominance was given by Carl Correns. Next line. This phenomena, it is a very important point that you must know, no need to write in the board's exam. This phenomena with respect to flower color, same thing, with respect to flower color, the same, everything is same, can also be seen in Mirabilis jalapa, in bracket 4 o'clock plant. Okay, but in the board's exam you are not writing Mirabilis jalapa, you are writing only which one, the dog flower or the snapdragon. But remember, even in Mirabilis jalapa, also called the 4 o'clock plant, where if you consider the flowers, they are available in red and white. When you cross these true breeding plant in this plant also, you will find what? Pink color or the incomplete dominance phenomena. Clear? Next line in the note, a separate point. In that only next line, in incomplete dominance, phenotypic ratio is equals to genotypic ratio. Phenotypic ratio is equals to genotypic ratio. This is one of the very important observable feature. Do you remember this? In which condition we saw the phenotypic ratio was equal to genotypic ratio? That was in monohybrid test cross, the previous session, right? Monohybrid test cross. If the given F1 plant is heterozygous, in that case you got what? Phenotypic ratio is equals to genotypic ratio. ಆ ರೀತಿ ನಿಮಗೆ ಟೆಸ್ಟ್ ಕ್ರಾಸ್ ಅಲ್ಲಿ ಸಿಕ್ಕಿದ್ರೆ ಅದಕ್ಕೆ ಒಂದು ಮೀನಿಂಗ್ ಇತ್ತು. ಇಫ್ ಬೋತ್ ಆರ್ ಈಕ್ವಲ್ ಯು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದ್ಯಾಟ್ ದ ಗಿವನ್ ಪ್ಲಾಂಟ್ ಇಸ್ ಹೆಟೆರೋಸೈಗಸ್ ಅಲ್ವಾ. ಆದರೆ ಇಲ್ಲಿ ಕೂಡ ಸೇಮ್ ಥಿಂಗ್ ಸಿಗ್ತಾ ಇದೆ, ಫಿನೋಟಿಪಿಕ್ ರೇಷಿಯೋ ಈಸ್ ಈಕ್ವಲ್ಸ್ ಟು ಜಿನೋಟಿಪಿಕ್ ರೇಷಿಯೋ. ಸರ್ ಏನಾದರೂ ಸಿಗ್ನಿಫಿಕೆನ್ಸ್ ಇದೆಯಾ? ನೋ. ನಾವು ಒಂದು ಥಿಂಗನ್ನು ನೋಡ್ಬೋದು ಅಷ್ಟೇ. ಇಟ್ ಈಸ್ ಅ ಅಬ್ಸರ್ವೇಬಲ್ ಫೀಚರ್. ಇಟ್ ಡಸಂಟ್ ರೈಟ್ ವಾಟ್ ಇಸ್ ದ್ಯಾಟ್ ಸಿಗ್ನಿಫೈ ಎನಿಥಿಂಗ್. ಇದರ ಯಾವುದೇ ಸ್ಪೇಷಲ್ ನೀವು ಮೀನಿಂಗನ್ನು ಥಿಂಕ್ ಮಾಡೋದು ಬೇಡ. ಅಬ್ಸರ್ವೇಬಲ್ ಫೀಚರ್ ನೀಟಲ್ಲಿ CET-ಯಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ಬೋದು. ಇನ್ ದ ಗಿವನ್ ಆಪ್ಷನ್ಸ್, ಇನ್ ವಿಚ್ ಕೇಸ್ ಆ್ಯಂಡ್ ಆಲ್ ವಿ ವಿಲ್ ಸಿ ಫಿನೋಟಿಪಿಕ್ ರೇಷಿಯೋ ಈಸ್ ಈಕ್ವಲ್ ಟು ಜಿನೋಟಿಪಿಕ್ ರೇಷಿಯೋ. ಈವನ್ ಇನ್ ಟೆಸ್ಟ್ ಕ್ರಾಸ್ ಆಸ್ ವೆಲ್ ಆಸ್ ಇನ್ ದಿಸ್ ಕೇಸ್ ಆಲ್ಸೋ ವಿ ಸಿ ದ ಸೇಮ್ ಫಿನಾಮಿನಾ. Clear? This is also very important, ಗೊತ್ತಿರಲಿ. So here when as we get pink color, one thing is clear, it is not obeying law of dominance. Phenotypic ratio is also not same as the Mendel's monohybrid cross phenotypic ratio. So remember student, it is an deviation or exception to what? Law of dominance, and this exception is called incomplete dominance, shown by I have given you 2 examples. Many more examples are there that we will discuss in the need session. The remaining 3 exceptions are codominance, pleiotropy and polygenic inheritance. When you study these 3 also, remember that they are the exceptions. They don't follow Mendel's monohybrid cross, the concept called law of dominance.